Okay, boys and girls, we're here. I think we're live. For some reason, YouTube is saying live in 22 minutes, but I know that I have it set for 2 p.m., so I have no idea.
CST.
Yeah,
2 p.m.
2 p.m. CST, but it says 22 minutes.
In my time is 3 p.m.
Yeah, and I don't see it going live. December 30th, huh? For some reason, it hasn't hit YouTube yet, and I don't understand why. I don't know what's going on. Help me, Walker, help me.
Where are you guys? Come.
I know. Come on, guys.
Pop up.
I sent Walker. I'm live, but I'm not. Why am I not seeing it on? Oh, something froze. It froze. Huh. Google. Okay, we're live. Hey, Walker, do you want to come in? Yeah, I was live, but for some reason it was frozen on my Chrome browser, and I didn't get the usual blaring microphone. Anyway, okay, so we are here today to go over University of California in Santa Cruz browser, that one? The UCSC browser, is that the one you...
Let me show you first, and if you want, you can take over in your site if it's easier for you.
Or I can follow along.
Yeah.
So I have a protein for us.
First, I will go to open my browser. Let's open it in a new.
Oh my goodness.
New window probably will be. Okay, so So I'm coming here and try to share.
I'm gonna make you bigger.
Share.
Big.
Share.
Keep getting fuzzy.
Is there sharing right now?
Oh, it is, yeah. I'm sitting on my, like I say, I'm not awake yet. I'm trying.
Mm-hmm.
Do we have to show, I got it full screen because it'll be easier to see.
Yeah.
Is that okay?
And now I will just, let me see. So I'm just closing your windows and I'm in front of my windows, but how I'm at the moment, is there any problems right now? Everything
No.
Yeah. Good.
No,
So
I can see your mouse.
You see my Google search.
Yeah. Yep, and your mouse. So that's good.
Oh yeah, that's good. Okay, so just everybody go to UCSC Genome Browser. So first I want to say that there is many genomic browser. This is not the only one, but this is the one that I like most. And it was for many years my main browser for genome. So you see when I'm changing here the pages, right? So
Yep.
Cruz Genomic Institute Genome Browser. Now that is the main entrance here. What is all about here? Of course, you have lots of tools, and I can show you directly if you go in a blood, for example.
Tools. Okay.
For example, you see this blood. You can directly paste here a sequence that you want to look inside the genome.
Okay.
And also here on the left side, you can see a human or any kinds of genomes you can choose from. Even cats are here. All right. I'll go back. Here you can pick up the genome that you like to work with. There is several ways, of course, to pick up the proper genome, but let's say we would like human. Usually, so there is a different several human genomes, and I guarantee you this is just the last one, but there are many more. So you
38
can,
is the one.
yeah. So this is a human genome, just 38 it's named, and
Yep.
this is the different builds of the human genome. And the same is for mouse. For example, mus musculus 39. And even you have COVID-19 here.
Nice. So we can
Yeah,
see if we evolved from COVID.
yeah. So, and just pick up some. So my advice here, it's not necessary to have the latest buildup of genome to have accurate data.
Okay.
What I mean, it's the advantage of previous build is that they have more tracks to compare with. So, but let's go somewhere. Let me say human genome 19, for example. So I'll pick up this just for the heck of, but you can figure it out. I mean, here is a big data of many different species.
Okay.
Dogs, horse, Hawaiian. What's Hawaiian? Hawaiian monk seal. Okay.
So right away, let's see. Did you go to? Did you go to 19? Did you pick 19?
I didn't pick yet, so let's
okay.
pick it up. So I'm just showing that you have a lot of choice.
Many.
Many, yeah. So I pick up human genome 19. This is the build-up 19.
Okay.
So
what is it?
You see now?
What is it showing us? The first chromosome, or no? It's showing us
here. So yeah, I mean, okay. So first of all, here is the Zoom activity. You go in, go out. After that, you can enter a specific gene. For example, if you want to look just some gene, that is the place. Or you can look the entire chromosome. So particularly here, it's open chromosome 1, but you can give me another chromosome, whatever you want.
So where do you pick chromosome?
Here. So you can pick up in whatever you want.
Right.
Yeah. So on the go, see here the field. You can pick up just by gene or by coordinates or just entire chromosome. So let's take, I would like to have a chromosome. So you write down CHR.
Yeah.
And directly after that, pick up which chromosome. Let's say 2 or 19. I don't know. Which one you want? Tell me something.
Let's do 2.
Let's go to 2. Okay. So you click 2, chromosome 2. And immediately the system should recognize that you're looking for chromosome 2, entire chromosome. So here we go. We are right now in chromosome 2. Of course, as big as the chromosome is, as much in load.
Yep.
As slowly will be your internet.
Yeah. Mine came back already. Yours is waiting, right? And
now,
there are so many genes.
Yeah, there's a lot of genes. Sometimes you don't need that many genes.
Right.
But this is the entire chromosome 2. So you can see here everything that is chromosome 2 is in this site.
So I am
and
saying
all of
something different than you are.
Why that? So
I don't know. On let's see, what is wrong? I'm seeing the very first chromosome I have in the upper left. You have U2, and I have CMPK2. So why would we be different? We have,
Okay. Don't go in such a details. You will understand why. It depends how you choose the actual tracks.
okay.
So and I go. So you have a different tracks here. So what you have in your field is a different tracks. On the first track is the scale, of course, how big it is. So, and this is the scale. It shows it from here to there is 100 megabytes. Sorry, bases, not megabyte.
Okay.
Megabase. And after that, it depends what you load it as a tracks. So for example, here in my view, it's alternative haplotype, but you may have the genes. So this is the genes.
No, I have alt haplotype, and I'm looking at family 110C, and I don't understand why.
So just
You are scrolled over to the right because I see on top. I see U2. Okay, go back to the top again. This is the kind of stuff that these little details that get students very confused. But I'm looking at U2, but U2 for me is way over about one third of the way through the page, and U2, and I don't know. There's something
so
different.
I mean, to make it easier, so let's go just to, I will scroll down to show you the main features of the tool, and that's very important.
Okay.
So I mean, currently right now here is just the specific tracks. Don't pay attention exactly what is shown here right now.
Okay.
We'll do that a little bit later, but just for the moment, just overall to see what and how to work with this browser. I'm scrolling down. So of course, this is the genes in my tracks. You have to know that it depends how you'll set up whatever you want to see, that how you'll show. Meaning the different people can see different things. It depends what they can put as a track.
Yep.
So going down, many genes. And I'll show you when you go down to the specific genes what will happen. But now just overall the view.
So these are displayed in order from the upper left,
Yeah.
going down and then reading left to right.
Everything in molecular biology is DNA or RNA. It is shown when you have to display. It shows from 5' to 3' end.
Okay.
And that's very important.
Yep.
It doesn't matter what you're going to show. You have to be aware that it's from 5' to 3' end. And I'll show you how actually the genes could be in different directions, but still will be in 5' to 3' end. So here, when you go down, so we finish with the actual genes. And now there is different tracks. So for example, here I have a SNP track.
Go ahead. I'm sorry.
And also there is a different, so DNA is cluster. This is, for example, particularly in this particular track is about epigenetic marker like H3K27 acetylation. This is histone 3K27 acetylation. But you can change it. Everything is changeable here. This one particularly is kind of multiple alignments, conservative regions. Also, you have different levels of SNPs. Particularly here is the common SNPs and repeat masker, which it shows the repeats. However, some of the features of these tracks cannot be shown until you don't go to the specific level, the specific distance. So If you zoom in the genome, then you can see more details.
Okay.
Now, you have the ability to add your own track. So you can make your own track and you can load it on the browser here. And there is a different level of kind of skills to make your own track. But basically everything, it's about coordinates and about sequences, analysis of sequences. And now here's the actual tracks, and that's the very important moment. Here, this is something that you have to, it's very important. All right. So, based on this panel, you can tell actually the browser what actually you want to see. Because the features are so many features, a lot. You can not load everything. It's impossible for one simple computer. So
okay. What does it mean that they have hide on there? Does that mean it's already showing, or does that mean
hide you don't see them? You don't see this track. So every single square, it's a specific track, and just only few are shown here right now. So, for example, I will close. So here's the moment that you have to customize the tracks and whatever you want to see in the Genome browser.
Yep.
Because obviously you don't want to see everything. So let's go and customize for our purpose. So I will close these publications by saying hide. And once we customize here and refresh, now the picture will be different on the top.
Okay.
So whatever you can see. So let's go from the top to the bottom. First is the mapping and sequences. So base position, of course, we want to see. Fixed patches, I don't know exactly what it is. So now if you don't know what the actual track is, just click over the track here. For example, I don't know, for example, what is alternative haplotypes.
Okay.
Just let me say I don't know. What you have to do, you just click over and we'll go in another page in details what actually this track is all about. All right? Okay. And based on this, you can even manipulate from here and say, okay, I don't want to see that, so I'll put hide. And I'll go submit, and we'll go back to the loading.
And if you hit any of the refresh buttons, it'll pop it up, right? Let's see.
Yeah, that's the problem. I forgot that. So, the better way is just to start with what you want to see, and then you can go.
So one point of confusion for me right away is that when I go down and look at these track sequences, you know, the track control panel, I get a different order than you get. I have mine looks different than yours. Okay,
yeah, it depends on the build that you.
Okay.
So if you picked up different build, if you picked up, for example, Human Genome 32, not 19, for example, it's a different build, and the tracks are different for different builds.
Okay, so which track? Okay, repeat masker. I've got that one selected. Should I hide that one? Repeat masker?
Yeah, it's a good. So repeat masker is a good. So what I'm doing here, so from mapping and sequencing tracks, I don't see anything that right now I want to show you. But for example, I can show you GC%. Also, base position will be shown. I can show you GC%, and GC%. So there is 3 different levels. One it's a dense, another is full, and of course you have height. So dense means they will compress the information. It's easy to load. If you put full, it will be more spread it. So I'll put GC% here just to show you. So UCSC genes, it's a packed, but I will hide it at the moment. The genes by itself, it's a ref-seq. This is the genes that what you want to see.
Okay.
So I'll put it in pack at the moment. And I'll show you to manipulate the tracks from the top. So once you go on that. Phenotype and literature, you don't want to see right now anything from this, although you may want eventually. COVID-19, we don't want to see anything. mRNA and EST. So this is what it's all about, different mRNA sequence. Personally, I do not recommend to go easy on this. Just for the first, just don't load those, because there are so many sequenced variants that you can completely lost yourself.
Yeah.
Expression. I will close everything from expression. So you put height. Regulation. So here you may want something. And they are very different. For example, CPG Island, very interesting. So I'll put CPG Island, show. But I will close this encode regulation. However, you can go in a nucleosome, lamina. So nuclear lamin. So what is this major? So for example, you have a DNase track. So the people did experiment and they tried to do, for example, something like how sensitive is the chromatin if you add DNase and try to digest the chromatin with the DNase. And of course, as compacted is the chromatin, as difficult will be for the DNase to digest the chromatin. Therefore, this track shows how compact is the chromatin. So yeah.
Okay, I'm really interested in the one right below that nuclear lamina.
So nuclear lamina is the chip assay. So this is the chromatin immunoprecipitation where the nuclear lamina is interacting with the chromatin.
Yeah, that's that.
You can show this. So let's show this. Okay, nuclear lamina will show it.
Okay, and that is under regulation, right? Okay, let me see.
And the regulation.
Okay.
You have enormous. So comparative genome. This is for us when you try to debate with creationists or for the evolutionary purpose is the comparative genome. So you can show conservation, and I put here conservation in full. So everything that it's conservative will be shown. Okay, but you can specify, and you have ability if you go inside. For example, if you go inside in any of those tracks, you can additionally show exactly what you want. So the track by itself is a half a day. You can go inside and just to specify what exactly you want to see from the different tracks.
Yeah, I noticed I had conservation at full from before when I played with this before. So
I mean, you can have conservation only with mammalians, or only with dogs, only with cats, or only with primates. It's up to you. So you can specify what kind of conservation. For example, here down you have a primate chain net. So this is another level of comparative genomics. So you can see exactly with the chimp and all the others how it fits very well, or doesn't fit. I'm not going to switch on this, but it's up to you. If you go down, so I see another topic of tracks, which is the Neanderthal assembly. Isn't
nice.
cool? That's a cool.
Yeah.
Right now, I'm not going to switch on, but you can play, of course. So you have so many things to play with. And you can go in Neanderthal sequence and you can show or not Neanderthal methylation. Oh my goodness.
Yeah.
SNPs.
They have Denisova on mine shows Denisova too.
Mine also show. Yeah. You can do Denisova assembly analysis.
Is Walker still in chat? Oh yes, there you go, Walker. You should be. You should come in and talk to us if you're feeling up to it.
Variations. This is another set of tracks, and again, only the double strand SNPs. So I will close these SNPs. I will put just something like common SNPs. There are different levels of SNPs. And don't forget, every single SNP is a different percentage. So let me say, if you take 1,000 people for specific SNP, those SNP might appear only 0.5% of those people, or maybe 60% of the SNPs. And this is a very useful tool for all of the forensic scientists. Because, as you know, half of the genome comes from the mother side and the another half comes from the father side. Correct? But they came with different SNPs. So the SNPs from one side, this is a single nucleotide polymorphism. That means SNP. So, and from one side, it's coming one set of SNPs, and from another side comes a different set of SNPs. And once you go in forensic science and sequence a specific piece, and if you choose a particular piece which is specific, very variable, you can figure it out even who the person is in this DNA that you sequenced.
Okay.
So this is about SNPs. I'll put just common SNPs.
Okay.
And I'll put it as a dense because I don't want to spread it too much.
Okay.
Just to show, to demonstrate it. Repeat masker, this is about the how many, so it's just about repeats.
Oh, so microsatellites.
Yeah, different repeats. And repeat master actually, in fact, is almost, it's a very good set of repeats and show you difference about it. So once you go in a setting of all of the tracks that you want to see, just refresh it. It doesn't matter where you will refresh it. So just click somewhere, refresh. I'm clicking refresh here and waiting at the moment. So of course you have to.
I'm still seeing the. Oh, there you go. Oh, yeah, mine is.
Yeah, you have to wait.
Why am I still seeing all of the? Let's see, not just see. I'm seeing all the genes yet, and you're not, or are you? And I couldn't find CPG Island.
Oh, did you put CPG in your place?
I think I cut out. What do you cut out to get rid of all? Okay, all the genes. I hit them, but it didn't.
So
refresh again.
Let's some way in the chromosome. So now we want to actually see actual DNA. So we want to see the actual sites. So you pick up from the entire chromosome. So there is two ways. Once you set up your tracks, you can go either directly into the gene. It's not necessary to be chromosome 2, right? As we choose. Could be any. So you can give me a gene. Give me a
Okay.
gene.
K as in
Okay.
I don't know. Yeah. P as in Paul. N as in Nancy.
N
A
N A
as in Alpha. 1. Yeah. KPNA. That first one there.
K P N A. So this is
KPNA1.
just click from 3.
Yeah, it's a karyofurin. That's the important alpha that imports things into the nucleus. So
Okay, so I entered scene here. So what you can see now, I will go a little bit above, so I will zoom out a little bit. So see on the top you have a kind of guidance. Are you going to zoom out or zoom in? So zoom
okay,
out 3 times, so 10 times. So I'll go, for example, 10 times, zoom out from this gene, click it 10 times, and automatically will zoom out. Your gene will be there in the middle. So whatever you set at the moment, it's in the middle here. You can see it KPNA1. But
yeah,
also you can see the surroundings. Now, on the top, you have the scale. That's very important. You see how big it is. So we are talking for 200 KB,
yeah,
200 kilobase pairs. And also you have coordinates. The numbers are here. So virtually, if you sit down and write down exactly the spot of the chromosome 3, you know exactly where to go next time directly. So you can say, okay, my gene is in a chromosome 3, coming in a 122.0.0.0 or 122.1.0.0 and so on, you know. So by doing this, you can specify the coordinates. And next time, if you don't know, for example, the place, because it might not be the gene, it could be between the genes. Therefore, you can still specify the place where you want to go. And next time, just by your coordinate that you can write down, you can go directly into the spot. If you find something interesting between the genes and it's not specify this place in any way, you can do that. Now, you can go another way. So you can just drag a place where you want to go, like this. See how I'm highlighting?
okay. Drag and select. Okay,
And zoom in. Or you can not just zoom in, you can highlight the specific spot. So see, I'm dragging my arbitrary, whatever it is, place. And here we go. So we have here and a few surrounding genes. So what is the surrounding gene? It's a PARP9, PARP15, GTX3, whatever does it mean, I don't know. Now, let's go. Also, I'll show you if you have some tracks that you don't like. Here, you can just click with the right
and
button
hide.
of the mouse and hide. So you can read out of something that by chance you didn't put, for example, or you can
Okay.
change it. Or ability to change the order. So for example, this GC%, I want to put it at the end. Just grab it and put down. You can, for example, at the very bottom. Oops. However, the problem is that sometimes you can clone and will go you and will show you. So, but so you can rearrange the specific order how you want to see that. And now let's go to the exon intron area. So zoom in, it's one of the good ways. So zoom in is just by 3 times. I'll put 3 times zoom in. Also, you have move. So you can move on the left, on the right. So by just clicking these buttons here. So if I click move on the left, so just clicking on the left, it will shift it. See how it shifted? On the left, on the right, and it's all about what you want to see, basically. Now, let's go further down, zoom in. Okay. Also, you can, with hand, just by clicking over, with hand, you can move the entire field, right on left. It's like a PDF file, for example. So if you click over and just hold the left button, you hold it, you can drag it on the left or on the right.
Okay.
And here we go. This is your gene. And you have the exons here.
Yep. And that first one, 5' UTR maybe, is that?
So let me show you something else. So I'll go in again, even further down. And by going further down, you can go even to the nucleotide level.
Yeah.
So, but now you can see there is a very small errors going from right to the left. See that?
Yeah.
So here is the exons, where is the just this small, I don't know, small bars are the actual exons.
Yep.
Between this is the intron area.
Why the arrows going left or right to left?
Okay, that's very, very super important. That means the gene is reading from this way. So that means that the gene is actually, it's on the other side of the strand. So you have representation from 5' to 3' in one, only in a one chain of DNA. And if you go on the right, sometimes I'm wrong. Okay, if you go on the right, you will see that your gene further on the right. Okay, so you will see that the first exon is actually here on the right side.
Yeah. Okay.
And it starts to read from here to the other side. So I'm clicking on the left, going in the left. Here we go. So this is the last exon here. So this is the last exon, which will be spliced and will be put all together. Now, let's back to the first exon.
How did you get back to the first one?
Just move around. You have move on the right, on the left. So you can zoom out or zoom in. So there's many ways.
Let me zoom out here.
So you can zoom out 10 times or 3 times. Okay, I moved too much. So yeah, okay. I'll pick up just this region, which is the first exon gene that you want. And you see the CPG island here.
Yeah.
The CPG island is exactly where the first exon is here. So, and that is telling you something about. And also, because the GC% is very high here, you can see on the top, if you put ZPG. But also you can see some conservative regions, and here is the conservative regions on that. So you can figure it out how conservative is this particular region. Okay,
Nice.
so I'll be back. Just give me 1 second because too much coffee, right?
Okay. Okay.
How many people we are here?
I'm not sure. Let's see. Let me take a look.
I'll be back.
Oh no, I didn't save the chat, guys. Let's see. So I'm missing chat, so I have no idea what everybody's saying. Why can't homosexuals drive faster than 68 miles an hour? I guess I'm not missing a hell of a lot. What? I don't think I want to know. Okay, we got 10 people watching. We got 9 likes. One of you is a bastard. Let's see. Probably Dimiter. Standing for Truth should be watching because he might learn something. You still watching, Walker? You still out there? You're leaving me here alone? What if you're the not? Oh, I might be the non-like. Oh, hell. It was me. No, it wasn't. Anyway. You're still there. So the first problem I have with all of this is when I pop it up and I'm trying to go along with them, is that things are in slightly different orders, and I'm ending up with slightly different zoom effects. And that always becomes confusing when you're trying to follow along on a video on YouTube for these genome browsers, is that there seem to be variations. And but right now, Dimiter and I are both using exactly the same sequence, and yet I'm seeing a different screen. I don't know why. And I think, let me see here, we have HG tracks dB equals HG 19. He's on 19. Last vert mode. Huh, interesting. So that always becomes confusing when you're trying to learn this stuff, is trying to find something. Like I could not find GPC islands when I did a search on this thing, and that concerns me. Maybe that's expression. Oh, I know what I might have done.
Okay, I'm here. So
I was just saying that I can't find GPC islands on my tracks.
CPG islands?
Or yes, CPG islands, yeah.
Well, just browse down.
Mapping, what heading would it be? Okay, oh, okay, I believe, let's see, Jeep. So,
where was CPG islands?
Which heading was it under? Well, they even have the Ellen brain map on here.
Okay, I will hide this one. Base GC percent. I will eliminate this GC percent. We don't need it. Let's find the CPG island.
Mapping and sequence. Okay, GPs.
Common SMPs. What is this? I just put it before a few minutes.
Yeah, I thought I saw it too. Oh, there they are. CPG islands under regulation on the right.
Yeah. So, yeah, exactly. Yeah.
Regulation. I do not have as many things as you do under regulation. I only have let's see, under regulate. Oh, wait a minute. No, oh, I found it. Okay, I found it.
Did you?
It's just so hard to find everything. CPG.
Oh, yeah, there are lots of features.
Yeah.
I prefer usually I prefer a little bit not the newest build-up,
Yeah.
because when you have a newest build-up, the tracks are not that many.
Yeah.
So all the tracks goes with the build-up.
So how do you
So
pick the version that you're using? Okay. I'm using HG19 too, but my display looks different than yours. So you must have customized your display somewhat.
I don't know how I did it. That I mean,
I can show you mine later, and we could go over this again. But
yeah.
that becomes confusing for students when the teacher's showing you doesn't match what you're getting on your browser. So
I mean, I didn't in some probably from previous time when I worked with this particular.
yeah.
Sometimes the best way is not to load too many things,
Yeah.
because if you load too many things, usually it's kind of confusing.
Yep.
Now I lost because I closed it. So let's put it back.
Trying to find my.
So if you go in full refseq, where you can see refseq.
Yep.
NCBI refseq. This is your genes. Also, you have UCSC genes, but this is additional.
I didn't have mine. Mine wasn't full. So.
Oh.
And then you shut off.
There is a different level. Yeah.
So let me try refreshing again and see if I can get where you're at.
So you can have full, you can have packed, and this is a different ways of display the same thing. Sometimes they prefer to be packed just to show it as a very small area, but not everything. So I will refresh mine and wait. Maybe I'm in a different regions. So if I zoom out.
Do I repeat masker? I'm trying to find. Maybe I didn't pay my bill.
Okay, so I will go back to UCSC genes. Let's try to put that back.
Okay, UCSC genes.
Yeah.
Okay. Did you select full?
Yes, but when the full, it's a good when you are not zoomed too much.
Yep.
If you zoom to bigger area, full will be very difficult to load everything.
Yep.
And also, for example, you also can see the different alternative forms, because what you can see here is the mRNA actually sequence to the genome. You see the same gene in different variants.
Okay.
Let's zoom inside the gene.
So that has a variable splicing, basically. The first one is shorter.
But you see so this is the KPNA1. But you see the difference. So you can see exon 1, 2, 3, 4, 5, but 6 is missing. Yeah. And after that, you have 7, 8, 9, and blah, blah, blah to the end. So now let's, so this is just demonstration to see the actual gene, but let's see what will happen if you go much deeper. So let's say I will pick up the last exon here.
Okay.
And I will go in the last exon, zoom in.
Okay, let me see if I can. Okay, so.
And you see the vertebrate conservative. So let's look, there is a conservative regions. So see how this is how conservative is the region.
Okay, so it's got a percentage of conservation. So,
Yep.
so
And you see clearly, for example, that this particular is exactly here. But between exons, it's not much conservative.
right. Yeah, so that means those are the we're not going to call them junk, but the junkyard, the I call it I call it evolution's laboratory instead of junk DNA.
Yeah. So you're closer. See, this is at the moment in my browser, this is 5K base pairs. 5,000 base pairs is this distance on the top. So you have a scale. Also, you're much further inside. Also the repeat masker. So the repeat masker at the moment is just dense, but I'll put it full, my repeat masker. And that will show you the different types of repeats. So one of the virus-like repeats like sine, line, LTRs, simple repeats, satellites, all of that are here.
Oh, nice.
And you see exact exon is the last exon. There's no much repeats there. So they're just arranged around, but not within the actual exon.
Yeah. Yeah.
And also you can see the curvature.
They're in the
example, yeah. You can customize with what you want to be, compare with what kind of species you can compare how conservative is this region. And right now in front of me, in my customized situation is res mouse, dog, elephant, chicken, and even zebrafish. So you can see that this particular, I can tell you right now that the particular regions where the highly conservative is the most important region. And if you delete it out, the organism might have a big trouble.
Yeah, no, Eugene Kunnev, that's what Eugene Kunnev calls meaningful information. The more highly conserved, the more meaningful the information is. So,
I mean, simply that means that it's execute vital function, something which is important. And you can, if you go and try to look right, for example, I'll just go on the right. See the conservative regions, how match exactly the exons area.
Yep.
You can see directly in front of you. That means something, right?
Yeah, it does.
So let's do something else. So, before going, okay. First, let's go to the level of nucleotides. I will show you also how to download the DNA.
Okay,
oh boy.
Where are you exactly? You're over on the right.
I clicked.
Oh, if you click, what do you call it? How do you go to the nucleotides? There's a way to go to the nucleotides up on top, isn't there?
Yeah, you can go zoom in the base level. Zoom directly into the base. Yeah. So you can click base and we'll zoom directly in the middle and you can see the bases. Now you have the actual sequence here.
No.
And you have
there's a way to get the protein too by clicking on one of these tracks. Do they have that feature on this where you can actually look at?
I think so, but you can translate. So for example, the translation is also there. You simply have to find the proper. So let me say, this is the UTR region. I think it's the 5' UTR. UTR mean untranslated region.
Yep. And that big exon on the left is an untranslated.
Not all. So
Not all.
it's translated. It will show you the amino acid.
Okay.
So and in fact, I don't know where I am within the gene right now. So I have to zoom out, see exactly which gene it is, and
think
I
you're
can
under.
figure it out.
Yeah, and you got to go. Okay, so what did you zoom out to?
So this is some kind of exon, but which one? I don't know right now. So I
Right.
zoom out 10 times, for example. Let's see what happens if I zoom out.
Oh my.
It's still, yeah. So
Still big.
zoom out.
Okay. Oh, depending on what. So that's 10 times where you're at. So that's why I was getting differences.
Yeah.
Yeah.
That's a very cool. You can. I mean, this is just a fraction of what you can do. The very cool thing is you can even, let me say you have sequence from somewhere. Doesn't matter, by your research, for example, you may have sequence. You can go here, go into the tools, go in a blot, paste there, and you'll find it into the genome where it is this sequence. So let me say this is exon. I will zoom in this exon. I will go further down to the base level. And it depends what you choose by tracks. So you can customize. It could be translated or not. So here is just the base right now.
So we don't have a translation.
And it's in this particular region because it's exon. And also you can see how well it conserves. So if you see the other sequences, it is highly conservative.
Go back to base. Let me zoom on an exon. I don't know which exon you zoomed on, but let's see here.
So, and this is 20 bases, the scale. See it on the top?
Yep.
Just 20 bases. And this is the site where the lamin B1 is attached. But there is many of those. I can tell you something else. Based on my experience, the lamin sites usually outside, not too much onto the exons area, on the genes area. Lamins likes not the genes. They just like to avoid the genes and to attach something that it's not genes.
Yeah.
That's in
that's what creates those these territories in the nucleus. The lamins, I believe.
Yeah.
They will kind of bunch things up around the outer membrane, the membrane of the nucleus, and
so let's see some transcription factors. So I'll go down to my tracks and see, can I find something that is transcription factors. So gene predictions, no, not that. Phenotype and literature, not. COVID, not. mRNA, no. Regulation, maybe, yeah. So regulation you have here. Oh, there is a encode methylation track, which you can see. A replication chip is something that I was working before. It's about how early and how late is this particular site in order when it starts the replication within the S phase. But we are not interested right now on this. You have also tissue specificity expression. You may see the expression of the tissue. Tissue specific MI. This is also for the miRNA. So once we close here, you can play with this browser with not just days, with months, and you still will find more things there.
Yeah,
A lot
yeah.
of more things. So transcription factor. So let's click on this TFBS. So factor binding sites. Here we go. So we would like to put a squash, for example, just to be there. And also you can specify what actually you would like to see. And also there is a very clear description how they calculated everything.
Yeah.
All right. I like this. So let's squash it and submit.
And if I hit refresh on mine. Okay, so now where did that go? What was that called again?
TFBS.
Okay, where did you where do you see it?
There.
I'm here.
So you might not see if it's not there. I mean, let's go zoom out 100 times out. And see what kind of transcription factors might be. Even more. So zoom out even more.
Zoom on another. The second one you did 100 again.
100. Oh, that's far away. 200. See transcription for CPG islands. This is lamin. Here's the transcription factors. So because
okay,
I choose to be squashed just to show you how it is. So I will put the transcription factor above, just the top. You just take and drag it. And let's go to see here. I think this is the area which is important for regulation. For some reason, this gene is regulated by the end of the gene, not by the beginning of the gene. I never seen that before.
Oh boy,
it seems to me that where those transcription factors? Here we go. So the transcription factor, I can put it in a full to see exactly what it is.
Oh, okay. Nice.
Here we go. So this is transcription factor binding sites. What does it mean? On the left, you see the something which is probably about transcription factors. And you can see what kind of transcription factors actually bind to this particular site.
A whole bunch of them.
Bunch of them, a lot. So I'll zoom even further down. For some reason, this gene, it's very important. This, whatever we see, is the 3' UTR. So this is the last exon, which is the 3' UTR, and you can see how many transcription factors are loaded on this. Very strange gene. Meaning strange that it looks like that most of the regulation comes to the 3' end, not to the 5' end.
Yeah.
Usually in the 5' end, but this one is very strange. I don't know why. It's interesting, but
so reading, okay, because we're reading this from the right to the left, is that the last exon you call the 3'?
Yes.
Okay.
Yes.
So you reverse it.
The transcription starts from right to left.
Yeah.
In this particular gene, if you go in another gene, might be from left to right. And usually, because in the browser, they show you the sequence only in the one strand of the DNA, not on the two of the strand.
Yep.
The other one, it's reading from the opposite side.
So if we were to try to find the, what the hell, the ATG, the start codon.
Well, you have to go at the beginning of the gene. So from the right side, for this particular gene, from the right side. So let's make another exercise for your purpose for ATG. I'll zoom out again 100 times. Oh, 100 times is too much. Oh, I forgot that I'm in a 2K. Okay. So I will pick up the very first exons here.
Okay.
Here. So I'm going at the beginning. It is quite possible. So to me, this is the first exon here. So I will go inside further. See the CPG island here?
Yeah.
I'll go even further. I want to mention something else. It's not necessary your first exon to have ATG, starting codon.
Yeah, I know.
But it should be 5' untranslated region there.
Yeah.
So
if it exists.
yeah. If I go inside, so let's go further inside. I'll go to base level now.
Maybe you're gonna have to
So
scroll.
you have to be also you have to understand that in this particular case, ATG should be written right to the left.
Yeah, reversed.
So you have to look something like G
ATG.
GTA.
GTA. Yeah.
Yeah, should look for GTA here somewhere. I recognize it in this way. I have to go down and look specifically in the track how to put such a track that will show the expressions, the transcription. I mean, basically, you have to see the amino acids. And I'm sure you can do that. Let's see how you
usually the green tracks are the ones that no, I'm sorry. But on
the green track here is Island.
Yeah, on NCBI it was the green tracks that you could expand and you could find proteins or amino acids.
You can do that. So you can cross-reference this data from another data which is in the NCBI, and you can see exactly the sequence. Also from NCBI, you can take the sequence, put it here in the tools.
Right.
Go in the tools, go in the blood, just paste it here, the sequence, and you'll get exactly here where it is. So it's not a problem to find exactly where is the UTR and where it's starting the translation, where it starts. This is not a problem at all. You just need to customize your tracks for that. Also, you can, for example, you can look very short repeats. You can look for, you can type some sequence and you can look at, can you find this sequence inside the genome? So where was that?
Oh my goodness, alt haplotypes. Hide.
So it's a track search. So track search, if you click track search.
Where did you find that?
It was just below the window, the big window.
Under
Well,
tool?
going back. So here, no. Autumn, you have a customize. You can add your customize truck. Yeah.
Got it.
So there is a truck search here. And if you click on the truck search, you can find sequence which you just looking. For example, G, G, T, T, A. Let me say something like that, short. And you can hit search. Not found. Okay. So, I mean, something else. So, C, nah, not found. Okay. So, you can look for a particular search. So go back to advanced. Okay, so there is advanced search. So you can find different types of specific sequence by whatever you want. I actually didn't investigate too much that.
Yeah.
So, and of course, everywhere there is a huge help. Okay, so to make the situation easier, so I'll put here the repeat masker dense, the actual conservative regions, okay. And also the genes, I'll put it in dense, the genes. And I will go to show you the Y chromosome.
Okay.
So C H R Y. See what will happen. Here's the Y chromosome. And here's the genes. Now pay attention that the Y chromosome, in fact, okay, let's pack the transcription factors also because it's too dense. And this is also, I'll pack it. The CPG islands also will be packed. See how the Y chromosome is only on the left side, is with the gene. On the right side, there's not much genes here.
Right.
So one unique features where on the right side, you may have a lots of different. So if we go in a region, there is no even one single gene here, for example. And let's investigate this one. No genes, not important.
Yeah.
So you go there, go down. Oh, I go up. Oh, sorry. Go here. In. I'll go to 10 times down. And directly I'll go to the base down. So on this side, you even don't have a good sequences. It's not sequenced well. Why?
Why is that?
Why that happened? The reason is the repeats. There's so many repeats. You cannot figure it out exactly how to align all of these sequences.
Yep. So the ends mean that for the audience that there is no.
It's nucleotide. Basically mean nucleotide, but we don't know. Yeah. If you go on the other side,
so all these comparisons they're doing with the Y chromosome and the stuff that Jensen is doing, I think he depends heavily on some of this stuff. I don't know what he's I don't know what he's doing exactly.
So you can drag over the chromosome and go directly into this particular place if you drag. So here we go. So we're on the left side of the chromosome, and there is the genes. You see the genes here, and you see also the conservative regions and so on. Also, if you go further, let me say I'll pick up one gene of those. Which one? So let's take something, I don't know what. And I'll show you how to download the sequence.
Okay.
So let's take this particular, I don't know how it's called, this gene, what kind of gene is, just pick up something. Maybe it's responsible for the male muscular or something like that.
Yeah.
Enhancement of the strength.
Probably be responsible for the part that makes males pissed off continually.
Yep. So let's see what kind of gene is this. I'll put full. It's a called gene PCDH11Y. What does it mean? I don't know. But so, and this is 200 KB. It's too much, but I'll show you just this region here. How to download the sequence of this region. So you pick up a region that it's in your interest. You like this region. You want to play with the sequence of this region, right?
Yep.
So you go, first of all, the sequence that will be downloaded will correspond to the window that you can see here.
Okay. That I wasn't sure of before. Yeah.
Now you go in the tools, and also when you go in the tools, in the downloads. Wait a minute. Oh, view. Sorry. I was actually browsing until 3 years ago this browser. I haven't browsed this, and they constantly change something. So this is a dynamic area. So you go in a view, and you go in a DNA. Also, you can make a picture of. So to download a specific picture, you can have a very nice, clear picture to use for a presentation.
Okay.
It doesn't matter what it's shown in the window. You can snapshot it very easily. So if you go in a DNA here and click DNA, first it shows on the top exactly this area that it's on the window that you can see.
Yep.
Also, you can specify, do you want some extra upstream or downstream extra bases? Sometimes you just want an extra 1000 base down, for example. You can put it here. And also there is a specific way to mark over the whatever you're going to download. So for example, there is a case color options. You can say here what exactly you want to see in your DNA.
Okay.
So for example, I want to see, when I'm downloading this DNA, I want to see the CPG islands. In order to do that, on a green, if I want to be green, if I want to be a red, just it doesn't matter. So the number is 255 here. 255 mean the maximum light, the maximum green.
Yeah.
But you can specify less green by putting less number. The number is smaller.
That's an RGB code for color. So.
Exactly. So you can have, for example, I would like the SNPs in red. I will take it 255. And also CPG islands, I would like to see in green. And I'll put 255 here.
Oh, you're one down. You got to go one up, don't you? Yeah.
Oh, yeah, you're right. Yeah, I put it in repeat masker. Okay. So 255, and I'll put 0 back here in a repeat masker. Here we go. And now, also, there is additional feature which you have to remember. Sometimes, if the gene is read it from right to left, so in the negative strand of the DNA,
Oh, you can reverse it.
You have to reverse complement it.
Right.
Because everything that you will see is 5' to 3'.
Yep.
So you have to reverse complement it in order to see. I don't remember this particular gene.
No, I don't either.
How was reading from left to right or right to left. I don't remember it. But let's say I think it is from left to right. So if it's from left to right reading, I don't need reverse complement.
Right.
But if it's from right to left, I have to click reverse complement.
Oh
And
my God.
now just submit it. And when you submit it, you see, and that sequence is color coded. Of course.
We're not getting any colors.
Well, there's no CPG, there's no those kinds of things. That's why you don't have it right now.
Yeah.
I can show you in another gene, for example.
We'll go back and do conservative or conserved and make that blue and see what happens. Maybe.
Okay, so let's try.
Okay,
so
make conservation, second one from the top.
Oh, okay, so that will make it in green. 5, so 0. Okay, let's see this way.
Let's see if we find some conserved tracks. Oh
Here
my
we
God. Go. Lots of it. Lots conserved.
So that is everything here is super conservative.
Yeah. Interesting. I did not know you could do this. This is awesome.
Oh, it is. It is. You just need time and computer.
Yeah,
and
well,
you can
you
play.
need lots of you need a bottle of Tylenol and maybe some aspirin too, because I'm telling you, after 3 hours, your head starts to really hurt bad.
Yeah, and 5x probably you need.
Maybe a quart of vodka might help some people, but I'm not allowed to drink that. So
just let me show you how to find into the genome something. So let's see. I will just copy this particular sequence here. Okay. Just copy. And now let me say I'm opening for the first time this browser.
Yeah.
And you go in a tool, go in a blot here and paste the sequence that I just copied, right? And also I'll specify human, okay. And this shows that it's HG19.
Yep.
And submit. So here it shows where your sequence is found. Also, it shows how many of your sequence that you pasted is found, because it might find not the entire sequence. I might find just the pieces of your sequence.
So it found some partials in chromosome 1. And
exactly. 2. So you're looking for something which is in a chromosome 3, 2, which is 100% identity, but it's not the entire sequence. So one, it's usually the higher score is on the top. So you see this sequence, it's in a chromosome Y from first to the last one, 100%. And actually, this is the place where we pick it up, right?
Yeah.
But also the same sequence you can find in the chromosome X.
I think it's amazing that's interesting. But it's also interesting that it's in 1, 2, and 3.
Yeah, and you can see a pieces in another, which is not everything, it's just small pieces of this sequence. So your sequence is 539 bases that you are looking, and sometimes they find only 20 bases that fit in a chromosome 3, for example, or 45 bases that fits in a chromosome 1.
So span on the end of it. So how big was your sequence? Was it about 539?
539 was the sequence. So let's go in the chromosome X. So we pick up from chromosome Y.
Yeah.
But the system found in the chromosome X 525, a little bit less. And I'll go in the browser. See here, you have details in the browser. So you click on the browser. And you're going in the chromosome X. And it will show you where is your sequence. So, your sequence is on the bottom. On the very bottom, it shows. Let's zoom out a little bit. So 10 times. Here is the sequence that we pasted. You see it?
Okay. Your sequence, yeah.
Yeah. The end with these red lines mean it is something that didn't fit very well.
Okay.
But mostly it fitted in 98% is identical. So this is just how you can investigate the genome, basically. You can do pretty much everything that you want. I'll go back to the one room for a second and I'll be back.
Okay.
So
where are my buddies? Why is nobody else in here? I invited a few of you. So you still out there, Walker? Somewhere? Not feeling well today? So if you were to debate Standing for Truth and he come up with some bullshit like he just recently come up with some bullshit that onions have a gigantic genome because onions can't walk and onions can't wear parkas. So they need this gigantic genome so that they can adapt to multiple environments in case a glacier comes or some fucking thing. But if you're debating somebody and you drag them down into this kind of detail to actually show you what they're talking about for mutations or whatever, it would be interesting. The debate would end with a huff. There'd be a rage quit. So. They do have a giant genome, but not because they can't walk and not because they can't wear overcoats, which was Standing for Truth's argument yesterday as to why they have, what is it, 5 times the genome that we have? Because they can't get up and put on a coat like we can, so they need a bigger genome in case it gets cold out.
Okay, I'm back.
Okay, we were just talking about Standing for Truth came out with an argument yesterday about why onions have a big genome. He says it's because they can't walk, and if it gets cold out, then they need that big genome in order. I'll tell you, that kid. Somebody needs to stop him.
Well, he might be right in some way. I don't
think so.
Let's investigate this hypothesis.
Well, I did go out and investigate this morning. Okay, I mean, even the craziest claims that he comes up with, I go out and I investigate it, and I found no evidence that the onion needs a larger genome because it might get cold, or whatever the hell he was trying to put out.
So let's go to CHR. CHR mean chromosome 22, for example. This is the smallest one. That's why it's easy to see. So this is a very
So that's the size of the chromosome,
sometimes, yeah, the entire chromosome.
51 million or 304,000.
I mean, 20 megabase pairs. It's almost so you can put the entire chromosome with about 40 megabase pairs. Actually, it's a 50 megabase pairs.
Yeah.
And this is the small chromosome. It's very famous for the trisomy of this chromosome and the Down syndrome. So
Okay.
if this particular chromosome is doubled or just tripled in the genome, there is a Down syndrome. So, and you can investigate, for example, what is there, right? It's easy. There's some genes which I was working with here, for example, MCM5, homeobox1 gene. It's very famous genes there. Homeobox XB4 or other genes. CHEK2, it's important gene for response of the DNA damage. MCM5 is responsible for the DNA replication. And many others, of course. This is just a few. So if you go inside, so I'll take something. I want to show you one more thing.
Can you pick one of those genes and
Yeah.
expand? Okay.
Yeah. So you can go, for example, take just area like this, for example.
Oh, okay.
Oh, whatever you want. I mean, you can just go directly into the gene. So let's take this one.
So on the left are those gene names on the left?
Yes.
Okay.
It depends how you show. I mean, how you want, because it's a full at the moment. So the display is a full, but you can have dense, you can have squash.
Okay. Yeah.
So that's kind of. So see, this is from this gene, which is SL20 SLC. Again, it's from right to the left, but here it's another one which is from the left to the right. Okay, so this is gene XPN. I don't know what it's all about, but it's obviously you have here the first exons of this gene. Sometimes they might have, see, this particular case, you have a CPG island, which belongs to two genes, one for the left and another for the right.
Yeah.
So cases are different. Also, you have transcription factors binding sites here. It is obvious also. And again, those transcription factors might belong to two genes from the left and from the right.
Right. Yeah.
If you want to download this, let's take, for example, this particular area.
Is there a way to download just the gene? Just the
Well, it's easy from the point of view of downloading. So it's kind of, you just go and drag only the gene.
Okay.
And after that, I'll show you. Just let's say that I would like to have this particular section to download. Go in the view in DNA as we discussed before. And go in a color coding. What is I would like to tell you. See the features that you can have with different colors, and also could be bold, could be italic.
Make the CPG islands blue, and
CPG
Let's see.
island will be blue. Okay. My point was, what I was wanted to, you remember this, the track name here that you see on the left corresponding to your tracks that you already browsed in.
Oh, right. So you would have more if you selected more. So
Exactly. That's what was my point. So if you want to see something, you have to have in your track, and you can color code it here. And then you will have it exactly the sequence. For example, if you want to see the actual gene, what is the exon introns? So it is genes here. So let's make it green. So the genes will be green. Common SNPs will be, I'll remove that, so just not to bother too much.
you probably want to remove conservation because that's green too. Or
Yeah, so you want CPG islands to be blue, right?
CPG islands, make them blue. Let's see
Okay,
what
I'll make it
happens.
blue.
Because I think we had some in that group, right? I'm not sure if you got them out or not.
So the labeling of exons, the exons will be green.
Where do you? Okay, I don't see exons.
It says genes. So it says US. The same will happen if you have NCBI ref sequence.
Oh my God, this could have saved so much time.
Absolutely. I mean, that's a fabulous. Also, what? Let me say red. I will put binding sites, transcription factor binding sites. So will be red. So I'll make them and hit submit. So this is a very short sequence that I picked up. It's 200 bases, basically. But you see the color coding.
What is that yellow?
Yellow is red plus.
Yellow was because it hit both, didn't it?
Yes.
Yeah. Okay.
So once you have mixed color in that case. So you have a red plus green, it makes yellow. So this particular area is a transcription factor binding site. You don't have CPG here because there is no blue here.
Yeah.
But that's how you can have it.
Oh, nice. This is wonderful. This would have saved me. So I did all this stuff. I went through base by base and picked stuff out for chromosome 2. And this would have helped me identify where the areas were. That's why
we can play here with, we can play here with days. There's so many features that you can study. The most important is your question. That's why I'm saying, if you know what you want, you can get it.
Right. Yeah.
So I'll stop sharing right now.
Okay. Yeah, we're coming up on 2 hours almost. We're about hour 40. So cool.
Any questions? Any questions from the guys around or something?
Let's see if anybody have any questions out there.
Walker,
I think Walker wasn't feeling good, so he took off. He's got it. So
well, it is recorded, right? So everybody can look it later. It helps a lot. If you can investigate. There's no limits.
Yeah. I was looking into those. What are they? Karyophyrins. They're the things that import things into the nucleus. And this morning I found a paper that they found homologs for those genes in archaea, in entirely different genes that don't have a nucleus. And so they found a lot of homologs going back, and it's really interesting to delve into the. You're looking at fossil evidence from like 3 billion years ago, which is pretty fascinating. So
yeah,
that's wonderful. Cool. for that.
Okay, Mike. I
Yeah,
mean,
that was very
and
informative. Wonderful. Thank you.
so you have to just play by your own. You know the principle, and there are lots of tools. I didn't explain to you. There's many more tools for that. So one I've been using, it's called in silico PCR. This is more practical in use.
Okay.
So you can, for example, paste your sequences for PCR, the primers,
Yep.
and you can look from this browser what will be your product from the PCR,
Yeah.
how big will be, and this is very also a kind of just practically useful. But there's many more, of course. There's
Yeah.
many more features. So first you have to find the right track, and second you have to know what you want to see.
Cool.
Uh,
Well, I'm going to poke around. There are a few things that I want to research, and I will have to ask you questions because I'll probably run into another blockage. but I think that is a wonderful explanation of this browser. Pretty cool.
so I can let me see. Can I show you another program just for a?
Yeah, there's also Uniprot and all that stuff where you can actually go out and get the structure of these proteins.
Yeah, I mean, also you can do alignments in some programs.
Like gene cards and
Gene card will give you the name of the genes, what you want to study. If you know the name of the gene, you can put it there and you can directly look. So you can look by name of the gene, you can look just by browsing, you can look just by pasting sequence.
yeah,
So you can take and many more features. So this is just the tip of the iceberg that I showed you.
yeah, that's probably something for a future episode. Maybe get into some of these gene or protein browsing databases. I was looking at one earlier, and I can't remember where I was exactly. Yeah, I typed in the wrong thing. I screwed up somehow. But anyway, okay, we can do that later. But you can also you can look at the 3D structures, and you can actually put your mouse over the 3D structure, and it'll show you the exact amino acid that you're on.
Well, this is about PDP database, so protein database. So you can go there and you can play with the actual 3D structure of the protein.
Yep.
Again, you must know what you're looking for and what exactly you're doing. But it is very cool also to see how the proteins are there. PDP database. Not the problem is that not everything is X-rayed, so crystallized to X-rayed and to figure it out the 3D structure. Not every single protein is already it's investigated, but a lot are actually. I mean, there are many that was done.
Yeah. Cool. Okay. Well,
Okay, guys, enjoy your time with browsing there, looking exactly into the genome, looking exactly in the sequence, and you don't even need to just virtually imagine the things. You can directly see exactly how it is.
and I'm sure if we look far enough, we'll find God somewhere in there.
Absolutely.
We just have to keep searching. Okay. Okay, we're going to end for the day. So
okay. Bye, guys. Bye,
everybody.
everybody.
That was your weather report today. So
Bye.
okay.